Well, thanks so much for the really kind introduction, Shashmita. I'm very excited to be here. I hope everyone online can hear me okay. And yeah, as Shashmita mentioned, my lab, we're a computational biology lab in the Center for Computational Biology in the Department of Biomedical Engineering at Hopkins. We call ourselves the JeffWorks Lab, and these are some of the friendly faces behind the work that you're about to see. And generally in my lab, when we look at a complex tissue, like say the mammalian brain, we're very interested in understanding how different cell types might be spatially organized in this tissue, and then how is this spatial organization potentially different in the context of health versus disease. And luckily for us, a lot of spatial omics technologies, particular spatial transcriptomics technologies, have now matured to really allow us to have the opportunity to begin evaluating these questions. And these technologies span many different flavors and brand names. And I realize, in this conference, not many have talked extensively about spatial transcriptomics so far. So for the rest of my talk, I really want to take this opportunity to teach everyone a little bit about what are these different types of spatial transcriptomics technologies, in particular imaging-based versus capture and sequencing-based technologies, and then ultimately, computationally, how do we go about analyzing these different types of data, and what are the things we should take into consideration or keep in mind. Great. So first, in terms of spatial transcriptomics by imaging, these technologies, again, Shashmita mentioned, include things like MERFISH. Generally, they're based on different variants of single molecule fluorescence in situ hybridization, where effectively, given an RNA with a known sequence, we can design complementary oligonucleotide probes. And then if the RNA is large enough and we can design enough probes or amplify the probe via fluorescent signal, we can effectively then image where the RNAs are inside a cell. And through many different styles of multiplexing, these different types of methods have different multiplexing and error-robust barcoding techniques. But effectively, what we have at the end of the day is the ability to, again, take a picture of all the molecules inside the cell. And I'm not sure if I don't think the pointer is working, but you can see that we can take pictures and distinguish that this molecule is gene A, and these molecules are gene B, and we can do this for hundreds to thousands of different RNA species that we target, we design in our targeted panel. Okay, and of course we can do this not only for one cell, but for hundreds to millions of cells in cultured tissues. Great.
Just give me the laser pointer, sorry.
Yeah, perfect. Oh, awesome, thank you. Great, so we can do this for hundreds to thousands of cells within fixed tissues and thin sections of tissues. So I hope that this allows you to appreciate how we really do have molecular resolution information. So therefore, we can actually begin characterizing, for example, a subcellular organization of RNA molecules within cells. I won't really dive into this much today because this is published work. But for example, we've previously been able to look at which RNAs co-localize, for example, with the endoplasmic reticulum versus different other subcellular structures. And this is, I think, a lot more interesting as we move to highly polarized cell types like neurons and begin asking questions like, well, what is the subcellular RNA composition of different dendrites, or dendrites versus axons, for example. We can also take advantage of subcellular organization of RNAs to begin inferring things like RNA velocity, in this case by distinguishing between nuclear versus cytoplasmic RNAs under the assumption that genes are born in the nucleus and will get exported into the cytoplasm where they can be translated into proteins. So by looking at disentangling the ratio of nuclear versus cytoplasmic RNAs, we can build differential equation models that allow us to try to predict whether genes are going to be putatively upregulated within a particular cell, and use that to project not only the observed transcriptional states onto a 2D embedding like a UMAP, but also the predicted future transcriptional states, and in that manner begin predicting temporal trajectories, for example. And again, this is published work, so I encourage you to check it out if you're interested in learning more. But today I'll really try to focus on things more at the cellular level. And as we heard yesterday from Dave Van Valen's group, you can segment these cells out using tools like Mesmer or cell segment anything now. But then we can count how many of each RNA species is in each cell, and that allows us to achieve a single cell resolution, effectively targeted spatial transcriptomic profiling, where now each point is a single cell. And we can see certain genes as being high in red or versus blue, low in blue for different genes that are in our targeted panel. For all the genes that are in our targeted panel. And of course, given this data, one thing that we might want to do downstream is, okay, now we want to, for example, normalize the data in order to accommodate for different capture volumes. Some cells are bigger than others, so of course they'll have more genes. Some cells maybe got sectioned and we only captured half of them, so then they have fewer genes. So we need to be able to normalize for that before we do, let's say, downstream differential expression analysis. And a lot of people, I think, understandably are porting over tools from single-cell RNA sequencing, which we've heard generally a lot from this conference. And I think that's a very reasonable thing to do at first glance, because of course these two types of technologies both give us single-cell resolution transcriptomic profiling. But I do want to keep to note, and for you to keep in mind, that at least currently imaging-based spatially resolved transcriptomics is a targeted technology, which means we are not at full transcriptome. And that has implications for downstream analysis. So my student Lila Atta decided to try to systematically demonstrate this by designing different imaging-based spatial transcriptomics gene panels for targeting the same particular tissue, and then normalizing the data using different types of single cell and bulk RNA sequencing normalization approaches, and showing how that can potentially impact our downstream analysis. In an ideal world, even if we have different gene panels, if I measure the same gene in gene panel 1 and gene panel 2, I should still be able to find that gene is, let's say, differentially upregulated in this particular cell type of interest. My results shouldn't change just because I changed my gene panel. However, so this is what Lila did. She designed different gene panels. In this case, she did something interesting. She picked gene panels that would focus on different brain regions of interest, which is something very reasonable, I think. If you're looking at cancer, perhaps you want to design a gene panel that focuses on immune cells. In the brain, we often do want to focus on only neurons, so we might design gene panels that skew towards that particular cell type of interest. So she designed different gene panels that would skew towards the ventricles or the habenula or the fiber tracts or the dentate gyrus. And then using these different gene panels, normalize the data using different count-based normalization, such as library size normalization, that is very common in single-cell RNA-seq analysis, versus DSeq2, which is more common in bulk RNA-seq, and versus something like cell volume normalization, and compared the differential expression of fold changes, log2 fold changes that you would obtain under the full gene panel versus a more skewed gene panel. And understandably, because we've skewed the gene panels to be particularly enriched in a particular cell type, now that the cells of that cell type are going to have a lot more genes detected just because we designed the gene panel to focus on that cell type. So therefore, the underlying kind of assumption of library size normalization that all cell types, on a full transcriptome level have roughly the same number of genes is going to be violated. And because we violate that assumption, now we get kind of funky results where, GPR75 is identified as significantly upregulated with a positive fold change in the full gene panel, but actually negatively or downregulated with a negative log2 fold change in the skewed gene panel. And of course, this type of discrepancy is not observed in cell volume-based normalizations because we're not relying on the number of genes that we detect in a cell volume normalization. But of course, there are other limitations to cell volume normalization that we try to highlight a bit more in our preprint. So I encourage you to check that out to learn more. In addition, you might imagine this type of issue will eventually go away as we scale up to a more full transcriptome measurement. So Lila used a simulation framework to show that if we have only a 50 gene panel that's very skewed, of course this has a much more pronounced effect than compared to if we have, let's say, a 5,000 gene panel, which much more closely approximates a full transcriptome measurement. So again, this is definitely something to keep in mind, I think, especially as these commercial vendors for spatial transcriptomics are now offering pre-designed small gene panels for us to use. So, just for you to keep in mind that it's important to normalize your data in a way that is going to be robust to these potential skews in your gene panel with respect to cell types. But assuming you normalize your data correctly and did everything well, or you designed your gene panel to be fully representative of the cell types in your data, well, now we can do things like things more akin to what we've done in single cell analysis with dimensionality reduction and clustering. And of course, in this case, because we have spatially resolved measurements, we can map these different cells back onto their original spatial positions and begin asking questions like, well, which cell types might be co-localized with each other. And of course, we can apply these technologies not only to mouse brains, but also human samples. So just as a demonstration or proof of principle, I wanted to highlight one example where we applied MERFISH, which is one type of spatially resolved imaging transcriptomic technology to characterize the spatial organization of transcriptionally distinct cancer subpopulations in glioblastoma. And in this case, we designed a gene. So Mario Suva's lab previously performed single-cell RNA sequencing on these glioblastoma tumors and identified 4 transcriptionally distinct neoplastic subpopulations. So we were able to use a single-cell RNA sequencing data set and design a gene panel of around 150 or so genes that specifically tries to distinguish between these 4 neoplastic subpopulations, along with other cell types in the tumor microenvironment, including oligodendrocytes, macrophages, and T cells. And again, we can now do the same type of dimensionality reduction and even co-clustering with the original single-cell RNA-seq data using tools like Harmony. But of course, now we have the spatial organization. So what my student Lila did was ask, within the, let's say, 30 nearest neighbors of mesenchymal GBM cells, do we see a particular enrichment of different cell types in the tumor microenvironment compared to what we might see if we just shuffle around the cell type labels? So given this type of permutation analysis, she was able to identify macrophages as being significantly enriched among the neighbors of the mesenchymal GBM neoplastic cells in a way that is not seen for oligodendrocytes, for example, and also not seen for different GBM subpopulations. And this ended up confirming quite well with some of the mouse models that Toshiharu is working on that suggested an interaction between GBM cells and macrophages to push actually GBM cells into a more mesenchymal state. So I hope this just highlights some of the potential applications of using spatial information to understand kind of cell-cell interactions and the reasons for these co-localizations in a tissue setting. And more recently, we've sought to formalize this type of co-localization relationship analysis. My students Rafael dos Santos Peixoto with Brendan Miller, a former postdoc in my lab, who's now at Merck, decided to create a computational framework for doing this type of cell type co-localization analysis, but in a particular way that is able to accommodate different length scales. And I think this is particularly important as these technologies scale up to, let's say, a whole organism setting. If we think about it, let's say we can do a whole organism human spatial transcriptomics. If we look at the whole organism setting, I could argue that, let's say, neurons and glia are co-localized with each other because they're co-localized because they're both present in the brain. So they're co-localized because of organ systems, and we have these different scales of organization, of course. So if I zoom in closer, perhaps neurons and glia actually form very different layers in the brain, or particular subtypes of neurons and particular subtypes of glia may form very distinct layers. So then you might actually interpret them as being spatially separated at a particular length scale. So this is all just to say I think it's important to be able to characterize these colocalization patterns at different length scales, which we show here using a simulation. Yeah, I hope you can appreciate that, for example, cell types B and C, the green and blue cells are kind of intermixed with each other, so they colocalize at a very fine length scale, whereas cell types A and C form these kind of circular functional tissue units. So they might not colocalize at a fine scale, but they do eventually colocalize at this coarser tissue scale resolution. So the way that we can quantify this is for each cell type, we can look among its nearest neighbors, but then compare that to a random permutation that's done at various different scales. So again, in this case, we are permuting the random background, but rather than permuting, let's say, all the cells in the whole tissue section, we actually only do a local permutation to accommodate for these different tissue structures. And that allows us to then identify, for example, in this case, cell types C and B are colocalized at a much finer scale if we look at the Z-score of that colocalization compared to cell types A and B, for example. And of course, we can apply this also to real tissues. In this case, this is a seq-FISH data set, another type of imaging-based spatial resolution or spatial transcriptomic technology, and be able to do this in a very fast pairwise manner to evaluate all the possible pairs for colocalization relationships. And in this case, we can identify, for example, endothelium and hematoendothelium progenitors as being co-localized at a very fine scale at the top. You can see they're very finely intermixed with each other versus other cell types that might be co-localized at a more coarser scale. And we can see that quantified via these trend plots again. So I think this is particularly exciting as we move forward. Again, you're welcome to check out the preprint just to learn more. But I think this is particularly exciting as we begin moving forward to be able to compare spatial organizational relationships in the context of health versus disease. And in this case, I should probably not call this disease because this is aging, which is a very mean thing to do for the older folks. But in this case, this is a thymus sample from a very young individual on the left. Each point is a cell. They're colored by different cell types. And on the right is a thymus sample from somebody not even that old, in their 20s. So you can just see this is somebody who's, I think, 6 compared to somebody in their 20s. You can just, I think, visually appreciate the spatial organizational changes that's occurring in the thymus. And this has, very important implications in terms of our ability to mount adaptive immune responses, especially as we continue aging. So I think moving forward, it'll be interesting to apply these tools to characterize the spatial organizations of these different tissues, particularly across health versus disease, but in this case, a temporal context in an aging context. And okay, so with that, I hope I've been able to show you how imaging-based spatially resolved transcriptomics technologies do provide us with molecular resolution transcriptomic profiling, which we can use to look at cool subcellular things, but we can also segment these cells, depending on the quality of our segmentation, into putative single-cell resolution transcriptomic profiling for targeted panels of hundreds of genes currently. And I hope I've been able to show you how normalization methods that have been developed for full transcriptome single-cell RNA-seq data may bias kind of downstream differential expression analyses, particularly if you've designed your gene panel in a skewed way to focus on a particular cell type, or designed your gene panel in a way that's not representative of all the cell types in your tissues. And finally, given the single cell resolution spatially labeled cell types, we can use tools like Crawdad to quantify cell type spatial relationships across length scales. Okay, so and now jumping forward to a different type of spatial transcriptomic technology that we in my lab call capture and sequencing based technologies. So these are generally, you might also hear terms like spatially resolved barcoding based technologies. And again, these technologies span various different brand names. The best known is probably the commercially available 10x Visium. But effectively, what these technologies all do is have some type of approach, typically via poly-T, to capture polyadenylated RNAs, and then through the cDNA conversion, tag that RNA with a spatially resolved barcode. In this case, for 10x Visium, we have these 55 micron spots that have been tagged with spatial barcodes. So therefore, when you sequence the subsequent cDNA, you can not only map the RNAs to the transcriptome and figure out which RNA it is, but you can also use the spatial barcode to figure out which 55 micron spot that RNA came from. So then what we get at the end of the day is spatial positions of all the spots, but then also all the genes that have been detected in that spot. So in this manner, we have a cell or a gene count matrix that corresponds to every spot in our tissue. So in this manner, we can achieve a full transcriptome profiling by sequencing, at least for polyadenylated RNAs putatively. In this case, again, I'm showing red as high gene expression and blue as low gene expression. But of course, we have all the genes that we've sequenced, not only just these two. And in this case, rather than single cells, we have these, again, we have these spots. So, again, how do we go about analyzing this kind of data? I think, again, it's very reasonable to begin porting over tools from single-cell RNA sequencing. In this case, we can do the same type of, let's say, dimensionality reduction and clustering analysis that I showed previously. But it's worth keeping in mind here that, yes, we are operating at the full transcriptome now, but we don't actually have single-cell resolution, at least as of yet. Maybe in the next couple of years, I've heard things coming down the pipeline. But at least for now, it's worth keeping in mind that these spots, again, there are 55 microns or so, which a cell is around 10 microns, depending on the organism and cell type. And therefore, each spot might comprise multiple cells, and therefore these multiple cells might represent multiple cell types. And therefore, the RNAs that you capture in each spot are going to also come from these multiple cell types. So why is that an issue? I think this is best demonstrated through simulations where we can, for example, take true single cells from single-cell RNA sequencing and mix them at different proportions to create an in silico kind of spatial transcriptomics data set that is actually the mixture of 3 different cell types. So here I've created a fake in silico, a spatial transcriptomics data set where some of the spots are comprised of 100% luminal cells, shown as these little pie charts in yellow, and then other spots that are going to be mixtures of parasites and luminal cells, and other spots that are going to be mixtures of parasites and macrophages. And of course, the gene expression that we get from these spots are also going to be mixed of these different cell types. So if we were to just perform, if we were regular transcriptional clustering, dimensionality reduction and clustering on this type of cell type mixtures, what we get is, in this case, 2 very distinct pixel clusters, which, even on a UMAP look, like 2 real distinct clusters, which we might be tempted to interpret as 2 different distinct cell types. But of course, in this case, because we simulated the ground truth, we know that actually pixel cluster 1 is a mixture of luminous cells and parasites, and pixel cluster 2 is a mixture of parasites and macrophages. And neither cluster actually is a real cell type in itself, and the transcriptional identity of this cluster is also going to be a mixture of these 2 different cell types. So therefore, in order to overcome this challenge, my lab developed a tool called STD-Convolve to recover cell type-specific gene expression and proportions within these multicellular pixels. And in particular, we wanted to do this in a way without relying on single-cell RNA-seq references. So this is published work, so I encourage you to check out the paper to learn more about the details. But just briefly, What SD-deconvolve does is builds on a machine learning algorithm called latent Dirichlet allocation that's been commonly applied in natural language processing to be able to estimate these cell type specific gene expression profiles and cell type proportions. In the context of natural language processing, LDA has been previously applied to do what's called topic modeling. So given a collection of lots of documents, for example, let's say all the articles in like New York Times, these different documents are ultimately comprised of words, and therefore we can think of topics as distributions over words, and we're trying to infer the underlying latent representation of these mixtures of topics within these documents. So therefore, you can think about if we have a document that talks a lot about, has words like bats and balls and teams, perhaps, those words, we should be able to see them covary across lots of different documents, and in this case, suggest an underlying latent topic about sports. Whereas if we see words like, spatial transcriptomics and single cell, especially if these words vary, if they co-occur a lot in one document and don't co-occur a lot in other documents, we can use that covariation across the documents to be able to infer that they must be about some similar underlying topic, and in this case, it's about science. So by the same analogy, I hope you can see that in the spatial transcriptomics setting, we have lots of spots now instead of documents, and each spot is comprised of lots of genes, and these genes are going to be, or the cell types are going to be distributions over these genes. So we're ultimately trying to infer the underlying latent proportions of these different cell types across these different spots, and also attribute these genes to these different cell types. So in that manner, when we apply this deconvolution to our simulation, again, we get out not only the deconvolved cell type proportions. In this case, we can deconvolve out 3 different topics or 3 different cell types and see that, proportionally, they actually match nearly perfectly with our simulation. So if I showed you the result, it would look kind of exactly like this, but we can further quantify that by looking at the correlation between the deconvolved cell type proportions and the ground truth cell type proportions. But of course, we also get out the deconvolved cell type gene expression profiles for each of the deconvolved cell types, which we can correlate with the original ground truth single cell gene expression profiles. And in this case, we have a very nice correlation as expected. Great. And of course, we can apply this to our real tissues as well. And in this case, I hope you can appreciate the difference with, let's say, a clustering-based analysis where, we essentially force cell type mixtures into distinct groups, whereas something like deconvolution can more appreciate the gradients of different cell types and the layer structures, fine layer structures within the mouse brain. And moving forward, we're particularly excited to apply these technologies and analysis tools to characterize gradients of immune cells surrounding glioblastomas in different mouse models. In this case, our collaborator, Mihalina Januszewska, injected immunocompetent mice with different glioblastoma cell line cells that have been modified to have different mutations. So now we can via deconvolution, not only identify the proportions of different immune cell type mixtures that are surrounding these tumors, but also began looking at their deconvolved transcriptional profiles and look to see how that might change depending on the different genetic background of the GBM cells. Okay, so for a summary of part 2, I hope I've been able to show you how capture and sequencing-based spatially resolved transcriptomics provides full transcriptome profiling, but currently at this coarse pixel resolution, where pixels might comprise multiple cells and therefore multiple cell types. Clustering analysis that's been designed for single cell resolution data when applied to this multicellular cell type data might be confounded by these cell type mixtures. However, deconvolution approaches using tools like SD-deconvolved and many others can potentially overcome this challenge and may be more appropriate for identifying cell type specific spatial patterns and gene expression profiles. Okay, so the last 5 minutes, I want to just now begin, draw your attention to, what happens when we begin applying these tools, technologies, not only to one sample, but many different tissue samples. How can we begin, for example, making molecular and cell type compositional comparisons at spatially matched structural locations? In this case, if I wanted to, for example, this is a kernel section of the mouse brain. Let's say if this is a healthy mouse brain versus this is a Alzheimer's model. If I wanted to ask, what is the gene expression at this particular region of the thalamus in the let's say sham or healthy versus the disease model, well, it's actually a little bit challenging to make that comparison because when you do your sectioning, when you do your tissue preparation, there's lots of diffeomorphisms that can be applied to the tissue, so it's not always easy to make that comparison. So to address this challenge, my lab developed a tool called ST-Align in collaboration actually with Daniel Tord, who's from UCLA. So we built on a lot of the work that Daniel did previously on large deformation diffeomorphic metric mapping and applied it to the spatial omics setting. And this is work co-led by my students Kaylin Clifton and Manju Anat. And what ST-Align does is, again, we can think of spatial transcriptomics data sets. In this case, we're doing a pairwise structural alignment between what we call a source and a target spatial transcriptomics data set, which we represent initially as XY coordinates of the cell centroids. So this is actually a computationally very intensive problem because each data set might have now millions of cells. So if you think about trying to align millions of points with millions of other points, it becomes a little bit tedious or computationally very intensive. So what we do is we convolve these single cell positions with a delta Dirac function and a Gaussian kernel to achieve a smooth functional representation of this data, which we can then, in this case, we can actually have a Dirac function that has an amplitude of 1 if a cell is present, or we can modulate that amplitude, for example, depending on whether a gene is expressed, whether for the first loadings on a PC. But in this case, we're primarily focusing on whether a cell is present or not. But effectively, this creates a smooth functional representation of the spatial transcriptomic data set, which we can then sample at a certain resolution to effectively create an image. So this is a process called rasterization. So now rather than having a million points, we have this smooth image representation of a source and a target image. And now this becomes a bit of an image registration problem where we're trying to find a particular diffeomorphism or particular transformation phi that when applied to the source image will make it look more approximate to the target image. And then this phi has a particular affine and a diffeomorphic component, which we can try to solve through a stochastic gradient descent by minimizing this error function, which has a regularization component and a matching component that I encourage you to check out the bioRxiv preprint and the online supplementary notes to learn more about the mathematical details. But I hope you can appreciate that, if we try to minimize this error term, then when phi makes our source image look more like the target, this will be minimized, and then this has to be subject to certain regularization constraints to make sure that we don't that we have a smooth diffeomorphism in a way that doesn't allow us to, for example, pick up one part of the tissue and completely rip it apart and move it to another part of the tissue to achieve the alignment. So this enforces a regularization that keeps our alignment realistic. So of course, after learning this transformation, we can actually then apply this transformation back onto our original XY coordinate of the single cell positions and move our single cells into this aligned coordinate space. So I think this is probably a lot more fun to watch happen. So I hope you can appreciate that in order to, again, achieve an alignment between our source and our target, we have to not only do rigid affine alignment or, like rotations and translations, but also much more finer kind of local diffeomorphisms to achieve this kind of structural alignment. And in this case, after we've aligned the source and the target, we can begin, for example, asking questions like, for this particular gene, looking across the tissue, what is the correspondence? And in this case, this is actually both healthy black 6 female mice. So indeed, we find a very high transcriptional correspondence for, all the genes assayed. And we can see that, in terms of these correlation plots. And I just want to highlight the difference between this versus if we were to have just done a rigid affine transformation. Now, our expression correspondence across space, it looks like there's some spots where in the target, it's not really expressed, but it's really highly expressed in the source. But this is actually just because we did a poor job with our alignment. So I think in order moving forward, it'll be important to try to disentangle the spatial molecular differences that are perhaps driven just based on, let's say, poor alignment versus something biologically interesting. And of course, we can also do this across technologies by aligning, our single cell resolution MERFISH to our Visium by leveraging the H&E images that correspond with the Visium. And in this case, we can also focus on just the region of the MERFISH that aligned with a high probability. And of course, now we can begin looking across technologies. I hope you can appreciate kind of the difference in terms of the detection of this particular gene, the spatial patterns that we're able to resolve across technologies when we have single cell versus spot-based resolution transcriptomic profiling. But of course, we can also, in this case, MERFISH is targeted and Visium is full transcriptome. So we can do, for example, single cell resolution clustering on the MERFISH data, but then deconvolution via SDDconvolve on the spot data. And even though, I would say that the detection sensitivity is quite different between these technologies, but ultimately they do allow us to resolve the same underlying cell type spatial organization, which is I guess good for science, that everything actually reproduces quite well, even across technologies, and in this case across animals and across different experimental conditions with fresh frozen versus FFPE. So we can align not only the brain, we can also align other types of tissues with high structural similarity, such as serial sections of breast cancer. And I think moving forward, it'll be particularly exciting to apply this in a cancer setting where we have serial sections that we can apply different types of, for example, spatial omics profiling tools. I think we heard a little bit in the earlier parts of the conference about epigenetics. So there are spatial epigenetic technologies currently being developed. Perhaps we can use one tissue section for spatial epigenetics or spatial attack, and one tissue section for spatial RNA-seq, and then use physical alignment as a way of integrating across these different data sets as a complement to some of the manifold alignment approaches that we heard about earlier in this conference. And I think other things that my lab's very excited about is, again, also applying these tools and technologies to a disease versus healthy setting. In this case, this is a great collaboration with Hamid Raab from the medical school at Hopkins, where his lab has developed these very nice, well-controlled mouse models of ischemic kidney injury. And we can induce kidney injuries for different durations and allow the mouse to recover and then sacrifice them, and look at the spatial distribution of different genes and cell types within the tissues. So the top is showing a sham kidney, and the bottom is a ischemic kidney. And in this case, for this particular gene, I hope you can appreciate, after alignment, we get a very nice correlation. However, there are other genes like this one that, in the sham has a very different spatial pattern compared to the ischemic kidney. And in this case, if we were to just do global differential expression analysis, the expression level of this gene is actually quite comparable across health versus disease. It's really the spatial organization that has changed. And through alignment, we can quickly be able to screen for all of the genes that fit this particular pattern. In this case, you can see how, there's no spatial correlation anymore. Great. So with that, I hope I've been able to show you how to, for example, make molecular and cell type compositional comparisons that match spatial locations. We can use tools like ST-Align to achieve a type of structural alignment across spatial transcriptomics datasets using diffeomorphic metric mapping. Additional details I didn't get a chance to talk about, but I hope you can check out the paper online bioRxiv to learn more, is how we can accommodate partial matches, and also we can also do 2D to 3D alignments, which is particularly nice in the context of the brain, where we often have sections that are not perfectly coronal, but always at an angle. I encourage you to check out the preprint to learn more. And of course, alignment across these spatial transcriptomics technologies can not only enable comparisons, let's say, across technologies for matched tissues, and potentially also integration of the benefits and of the strengths across technologies through this alignment-based approach, but then also potentially in the future are quite interested in 3D reconstruction and integration of multi-omics through spatial alignment. And with that, as Sushmita mentioned, my lab does believe it's very important to make our computational methods easy to use. So I encourage you, if you're interested in trying out these tools for your own data, to check out our lab website. And my students have made lots of nice tutorials. Our tools are installable via GitHub and Bioconductor as R packages, or via pip as Python packages. There are lots of tutorials to help you reproduce our analysis and also get started with your own data. So with that, I'd like to thank again my lab, our funding sources, and if you're interested in keeping in touch, feel free to reach out via email, and once in a blue moon, you can probably find me on social media. So with that, thank you, and happy to take any questions.
For several questions, if people can just please come up and ask questions from here.
Hi, wonderful talk. I'm curious with the alignment techniques, as the tissue starts to become more different from a disease, for example, do you worry that the alignment might erase from your ability to see it the actual biological difference? Is there like a human in the loop to monitor for something like that?
Yeah, absolutely. Yeah. So these alignments, I think with a lot of machine learning algorithms, it's like you can always apply it and always give you some solution. It's kind of up to the human to evaluate whether the solution is reasonable. So in our case, you can always morph something to look like something else, whether that morph is reasonable. I think, for us, we try to use actual transcriptional similarity as a gauge of that reasonableness. So, as I've shown you, most genes actually don't, after alignment, are quite spatially well correlated. They're only, it's a subset of genes that really change in their spatial patterning. So I would definitely recommend, sanity checking that most of, or at least some of your genes do still exhibit high spatial cross correlation after alignment. And of course, I think one thing we're very interested in is actually using the information of the diffeomorphism itself as a feature. Which parts of the tissue have to morph more in order to achieve this alignment. I think that could be other areas of kind of interesting spatial analysis that would be quite neat.
Hello, thank you for this amazing talk, and I really appreciate for the ST deconvolution method. So I have one question about this method. So how do you handle the collinearity problem for the similar cell type? Because we know it's kind of easy to separate immune cell, cancer, or stromal, but always like for example, if we want to separate CD4 T cell and CD8 T cell, they share a lot of common markers. So how this method handle this?
Yeah, that's an excellent question. Yeah, as you've hopefully others in the audience have noted, also, from the analogy that we really are relying on features that are going to be quite specific to a particular cell type or topic we're trying to deconvolve. So yeah, as the question noted, if you have collinearity, such as genes that are observed both in CD4 and CD8 T cells, those are indeed very difficult for us to deconvolve out. So typically we try to do a more two-stage deconvolution where at the end of the day, we need to feature select for the words or the features that are going to be relevant for our deconvolution. So in that particular case, we would actually try to remove out the genes that are common to CD4s and CD8s and focus on just the genes that are going to be more unique to each cell type or that we're trying to deconvolve out. So yeah, that's a great question and it's definitely a challenge.
Thank you so much.
Thanks for the great talk. And I have a question about how fast does the alignment algorithm converges and whether it's doable or realistic to apply it to align different time frames.
That's a really interesting question. So let's see, I guess generally we have a couple notebooks online that have time benchmarks to help you gauge. But so far in our experience, most alignments converge with on the order of minutes to hours, depending on how large the tissue section is and also how different they are. So for example, I remember, I think to align like this breast cancer section, because the alignment is mostly affine and not so much diffeomorphic, it took only a few seconds. Whereas for the brain, yeah, I think for this particular alignment, because there's a much larger amount of diffeomorphism, and I think it's a stochastic gradient descent, so it depends a little bit on how long you want it to run for, and also your descent step size. So I tend to be very conservative and choose very small step sizes, which probably makes things run a bit longer. But yeah, it definitely can take on the order of hours. But it's also optimized. So on the order of hours is for a CPU. If you do have access to a GPU, that will make things a lot faster also.
I see. Thank you.
Hi, great talk. So I think it's sort of more of a comment on the previous question about collinearity. For I have been doing text mining before, and I used an R package called STM. So unlike LDA, I think okay, it's been a while since after I learned LDA, so I might be wrong. I think in the Dirichlet model, the one topic increases in probability, that will be at the expense of other topics. Correct. But in STM, in contrast, in the model, the different topics can correlate. So I think that might help with the collinearity. Also, like in STM, you can also have covariates. I think at least for Visium, I'm pretty sure like usually the cell type the cell type compositions of spots are positively spatially have positive spatial R correlation. So I think can probably use like a spatial covariate. Possibly, I'm not sure. I haven't tested, but just a comment. I think.
Yeah, definitely. I think there's lots of opportunities for improving on these different tools, and yeah, I think there's lots of always good to sanity check yourself with different simulations. I think when we initially developed SDD-Align, I actually really wanted to use a hierarchical LDA because of this collinearity issue. You would expect, CD4 T cells and CD8 T cells to be hierarchically related. So arguably you would think that a hierarchical deconvolution would work better, but yeah, it didn't pan out in our simulations. So I think, if this is an area, I definitely encourage you and students in the audience to try it out. I think it's worth trying out. Yeah, great.